Welcome back to Bio 5, and today we're going to talk about gene expression. Learning outcomes are understand the structure and function of the genetic code, summarize the process of gene expression, describe the process of transcription, describe the process of translation, explain how gene expression may be regulated by a cell. DNA acts as a template for making RNA, which in turn acts as a template for manufacturing proteins. During gene expression, protein synthesis, transcription copies DNA into RNA, and in translation makes protein from the RNA. The structure and functions of proteins. Proteins are polymers composed of subunits called amino acids. There are 20 different kinds of amino acids to be found in most proteins, and the sequence of amino acids in a protein leads to a particular shape. Proteins have different functions in the body. They determine the structure and function of various cells in the body, and proteins are used as structural and regulatory components of cells. They are used as enzymes to catalyze chemical reactions, antibodies for the immune system, and hormones even in certain cells. Gene expression and overview. The first step in gene expression is transcription, when DNA template is copied into a strand of mRNA. The information from mRNA is then converted into protein in a process called translation, which is the second step. Over here is the central dogma of biology. You go from DNA to mRNA to protein, or polypeptide. The genetic code. A gene is a sequence of DNA that codes for a protein. mRNA contains triplet codes called codons, which are translated into amino acids using genetic code. The genetic code is nearly universal in all organisms. One of the codons, AUG, stands for the amino acid methionine, which is actually the signal for beginning any polypeptide. Most amino acids have more than one codon, and the redundancy offers protection against harmful mutations that can change the sequence of our bases. There are 61 codons corresponding to a specific amino acid, and the remaining 3 are stop codons. There are a total of 64 possible codons. Over here is a chart showing you all the different codons, the first base, the second base, and the third base. The redundancy in genetic code offers protection against possibly harmful mutations. During transcription, a segment of DNA serves as a template for the production of an RNA molecule, and all classes of RNA are formed by transcription. Transcription in the nucleus when an enzyme, RNA polymerase, opens up the DNA helix so that the complementary base pairings can occur. Then RNA polymerase joins the RNA nucleotides, and the mRNA molecule with a sequence of bases complementary to the DNA segment results. So there's a pairing between AU and CG. Here's a picture showing you that. Transcription of DNA into mRNA. Processing mRNA. DNA contains exons and introns, and before mRNA leaves the nucleus, the introns are excised so that only the exons are expressed. Here's a picture showing you going from the raw DNA to pre-mRNA and then to the final mRNA form. The primary mRNA contains exons and introns, and during processing, one end of the mRNA is capped by the addition of an altered guanine nucleotide. The other end is given a tail by the addition of multiple adenosine nucleotides. The introns are then removed and the exons are joined to form a mature mRNA molecule, and this is done by a complex known as spliceosome, which is composed of both RNA and protein. The RNA portion is not a protein, it's functioning as an enzyme, and therefore is called a ribozyme. Alternate mRNA splicing. Sometimes cells use certain exons. A single gene can result in different proteins, and small RNAs regulate not only mRNA processing, but also transcription and translation. Translation. During translation, tRNA molecules bring amino acids to ribosomes where polypeptide synthesis occurs. The ribosome consists of small and large subunits that will join together during translation and bind to mRNA strand. This creates a translational complex. The ribosome contains binding sites called A, P, and E. Here's a picture showing you an mRNA that is threaded between a ribosome of subunits, and polypeptide extends to the side. The ribosome contains binding sites called the A site for acceptor or rival, P site for polypeptide, and E site for exit, where the individual tRNA molecules can bind with the mRNA strand. The anticodon is a 3-base sequence on a tRNA that will complementary base pair with the codons of the mRNA. The complementary base pairing between the anticodon and codon is how the tRNA brings the correct amino acid into the correct order instructed by the mRNA strand. Each amino acid is coded by a specific codon. It has a tRNA molecule with a specific anticodon that carries it to the translation complex. The tRNA molecule is attached to the translation complex at the A site, where each anticodon pairs with a complementary codon. Here's a picture showing you that a tRNA delivers an amino acid to the ribosome. Upon arrival, its anticodon CUG will bind to its codon GAC. The tRNA molecule attached to the translational complex at the A site, where each anticodon pairs with the complementary codon. The order in which the tRNA molecules link to the ribosome is directed by the sequence of mRNA codons. The tRNA is attached to the growing polypeptide now and moves from the ribosome's A site down to the P site for polypeptide. Stages of translation. Translation requires 3 steps. During initiation, mRNA binds to the smaller of the 2 ribosomal subunits. Over here is a picture showing you that. You can see the machinery of the small ribosomal subunit, the large ribosomal subunit on top, and the E, P, and A site. Elongation is when the polypeptide lengthens, and 1 amino acid at a time. An incoming tRNA arrives at the A site. The ribosome moves down the mRNA strand 1 codon at a time, so that the P site is filled by the tRNA peptide complex. Over here is a picture showing you the moving of the assembly line inside this process. During termination, a spot codon sequence is reached, and the ribosome falls away from the mRNA molecule. The ribosome then dissociates and falls off. There are proteins called release factors which can bind to the stop codon and cleave the polypeptide from the last tRNA. Over here is a picture showing you that. A release factor that binds to that site causes it to break apart. Several ribosomes, called a polyribosome, can move along the 1 mRNA molecule at a time. And tRNAs are involved in bringing amino acids to the ribosome and binding to the codon with a complementary region called the anticodon. Here's a summary of transcription and translation. This is a chart that goes over what are the participants in the molecules inside this genetic expression. DNA, RNA polymerase, mRNA, tRNA, rRNA, and amino acid. A variety of mechanisms regulate gene expression, from pre-transcriptional access to the DNA to post-translational control of protein activity inside the cell. The mechanisms are pre-transcriptional control. DNA must be available to the enzyme necessary for transcription. Transcription control, a number of mechanisms regulate which genes are transcribed, and the rate at which transcription of the genes occur are very critical. Over here is a picture showing you transcriptional control and post-transcriptional control. Transcriptional control occurs as several mechanisms that regulate the genes that are transcribed and the rate at which they occur. That is, transcription factors, which can initiate transcription. Post-transcriptional control occurs in the nucleus after DNA is transcribed and mRNA is formed. Translation control occurs in cytoplasm after mRNA leaves the nucleus. MicroRNAs, miRNAs, bind to mRNA for inhibiting translation, and also the siRNAs can prevent translation by marking the mRNA for disruption by nucleases. Post-translational control can also happen in the cytoplasm after protein synthesis occurs. Transcription factors are DNA-binding proteins. After transcription factor binds to DNA, the RNA polymerase can attach to DNA and begin transcription. As cells mature, the differentiated become specialized, and specialization is determined when the genes are then active.